Hi, I'm Andrew Torr, and I'm going to give a quick overview of our paper on DeepSplit. So the overall goal of this work is accurate segmentation of dense cell cultures using convolutional neural networks. The key challenge is that common approaches such as UNet tend to overfit the foreground pixels, resulting in incorrect merging of neighboring cells. An example of this is shown on the right. The top image is the ground truth for one of the frames in our dataset, and the bottom image is the segmentation mask produced by UNet. To address this problem, we propose a multitask learning approach with several novel architectures, which significantly improve the separation of touching cells. Our multitask approach involves two tasks. The first is simply normal segmentation, while the second is a dedicated separation task, which explicitly learns the challenging boundary regions in the image. On the right, you can see an example of the reference data created for this separation task. Any pixels the task identifies as boundary pixels are set to black in the segmentation masks to produce the final combined results. The first of our multitask architectures is called Branchnet. This is essentially a UNet, shown in blue, with an extra series of convolutional blocks in orange on the end. The segmentation task output is taken from the end of the UNet as normal. The role of the extra convolutional blocks is to then convert the segmentation feature maps to a separation result, which is taken at the end of the network. Our second architecture is called double UNet, which is very similar to Branchnet, except instead of converting the segmentation feature maps to a separation output with just a few convolutional blocks, it uses a second, smaller UNet. Generally speaking, this network outputs superior results to Branchnet, as it has more layers available to specialize for the separation task, and the second UNet shape is able to effectively combine high-level context with localized features in a similar manner to a traditional UNet implementation. The downside of this architecture is that there are a lot more layers, so it takes a lot longer to train. Our last and most successful architecture is called DeepSplit. Once again, this involves a standard UNet, shown here in blue, with the segmentation output coming out of the end of it. But now we've added an additional decoder branch in orange, which handles the separation task. The most important feature to note is the cross connections from the segmentation task to the separation branch. These are critical as they provide context from the segmentation task to the separation. The need for context is consistently found to be an absolute requirement throughout all of our research. Initially, the separation task was trained using its own independent UNet. However, this really didn't provide satisfactory results at all. It transpires that without the additional information of having some idea of the segmentation map, it's very difficult to identify boundaries within the image. And really, DeepSplit's superior accuracy as compared to our other architectures is owed to the more extensive parameter sharing we see in this network. However, when we come to train these networks with a simple combined loss function, none perform as well as a single task UNet. This table lists the dissimilarity coefficients and average percentage errors for a selection of morphological features for all the cells in our dataset. The problem here is that for each of the multitask architectures, layers shared by the two tasks are being trained at cross purposes. The features required to get accurate segmentation results and accurate separation results appear to be actually quite different, making it very difficult to locate the global minimum while optimizing the two tasks simultaneously. These issues are resolved by developing a training schedule. First, the segmentation task is trained independently. This means that only the blue layers in the U shape will be trained, leaving all the orange layers with their random initializations. At the end of this stage, the segmentation results are saved. Second, both tasks are trained simultaneously, but with a low weight given to the segmentation task. This allows the separation task to be trained whilst being provided with useful context from the pre-trained segmentation layers. The separation results are then combined with the saved segmentation results from the first stage to produce final results, which comfortably outperform the single task ViewNet. In summary, this work proposed a novel multitask architecture, DeepSplit, which significantly improves segmentation results for dense cell cultures by adding a separation task, which learns the most challenging regions of the images. Our most interesting finding was that although context from the segmentation task is essential in training of the separation task, the two tasks are actually best addressed as independently as possible. We found improvements when we separated the objective into these two tasks, segmentation and separation, and we improved our results further by developing a training schedule where the two were trained fairly independently. Additionally, some more research we've done suggests there's quite a lot of mileage in an ensemble learning approach, where the training of the two tasks is further separated into two more independent networks,